Hi, this is Mike Edelhart, and I'm here with another edition of Inception, our podcast about beginnings, the beginnings of new companies, new ideas in science, sometimes even a little bit of a look at the future. And today, I think we may get some of all of that. So I'm here with Oriana Papin-Zoghbi from AOA Dx. Great to see you, at least Zoom style.
Likewise, great to be here.
So in this case, often I start these off by saying a little something about what the company does, but I think we may need to sort of help folks get a little bit of a sense of what your company does, because it's not something folks hear about every day. So maybe you should go first and explain to folks who don't know you and aren't investors what AOA Dx is all about.
Sure. At a high level, we're focused on the early diagnosis of ovarian cancer. Primarily, ovarian cancer is very challenging to diagnose. There are no tests available today other than a woman having to go to surgery and get a biopsy. The symptoms are not specific. It often takes months and months for a woman to get diagnosed. And unfortunately, 80% of the cases are diagnosed when a woman is already stage 3 and stage 4. Survival rate's only 28% at that point. So what we're working on is a blood-based test that is specific for ovarian cancer and sensitive enough to pick up disease at stage 1 and stage 2 as well, allowing a woman to get a diagnosis much sooner and be able to get to treatment faster. And women that are diagnosed stage 1 and stage 2 actually have a 90% survival rate.
I get it. And when we first talked and started going back and forth about this, I was flabbergasted to realize that there was no standard diagnosis. That it's basically you go in if you're a woman and you've got cramps or pains or sort of non-specific symptom, and it's like, well, we could send you home and maybe you'll die, or we can cut you open. And that just seemed like so 19th century to me. I was shocked.
Yeah, so were we. There hasn't been any innovation in this space in years. I think many reasons leading into this. A, historically, women's health is underinvested in. And B, it is a very, very challenging disease. It spreads very quickly. There are various different subtypes. And as you said, the symptoms are not specific. A woman has bloating, abdominal pain, cramping. You don't think cancer when you have those symptoms a lot of the time.
Right.
And especially amongst the premenopausal population, doctors, they don't have many options. It's not that they don't necessarily want to help, is that they don't know what to do. They don't have a test.
Right.
You can send her to surgery, but the risks of surgery are very high. So it ends up being this wait and see that doesn't lead to a lot of good results for women.
Right. So what you guys doing are a type of what's being called liquid biopsy, where you're trying to find an early, very specific indication, because one of the keys is it's not maybe yes, maybe no. The whole point is it is, and that doesn't require being cut open and all that. So how do you do that? What do you look for? What are you guys looking for? And do you know right now exactly what it is? Or are you looking at a number of things that might be it? And how are you going to go about finding it?
Oh, we know what we're looking for. These targets are called gangliosides. And essentially, they're lipid structures that live on the surface of tumor cells. And the family of gangliosides is quite large. There's about 40 different of them, and certain cancers present with certain types of those gangliosides. Think like a matrix, these cancers, these gangliosides. They've never been exploited before for diagnostic purposes. Nobody has ever tried to develop a blood test for them because they're so hard to extract out of the blood. And so the innovation that our scientific collaborator, Professor Segovia at McGill University, came up with was a method to extract them from blood, as well as proprietary antibodies to bind to them and detect to them. So the test itself, it's like a jigsaw puzzle. You have your target, you have your antibody, you need it to fit in perfectly so you can pick it up,
Right.
but only pick that up, like you said, specifically that and not something else as well.
Right.
And essentially that's what we're developing in an assay.
Got it. So you've got a one-to-one match sort of hook and receptor.
Yep.
that will allow you to grab these things and in some way pick them up. Now, once you grab them, how are you going to identify them? So are you going to, just sort of do physical analysis of the blood? Some of these things do phosphors, so they sort of glow if you get enough of them, or in some other way make clear the presence of these once you've
Yep.
hooked onto them. What about here?
In this case, we're using ELISA technology, and we're actually quantifying how much of them is present in the sample.
So
And
ELISA technology, for those who actually have like normal lives, is?
ELISA technology is an immunoassay technology. That's another big fancy word. Essentially, what you're looking at in this technology is utilizing antibodies to bind to a target, allowing you to essentially create a readout, a numerical readout, and then you can use those numbers to develop an algorithm. And so that allows us to see what the level is in background being normal, healthy as well, the levels in cancer, and then develop an algorithm that says, okay, above this threshold, then we read out a positive or a negative.
Right. Yeah, and so one way maybe, and correct me if I'm wrong, for folks maybe to think about this is if the doctor's feeling all over your body and would go, oh, there's a lump. This is sort of the statistical equivalent of that, which is normal, whoa.
Exactly.
And that's where it's a liquid biopsy. That looks like something that now justifies more exploration, more risk, more whatever.
Prevention. Exactly
And,
right.
right. And so where are you in this now? So that sounds like it'll be a helpful thing, very helpful thing to have available next Tuesday, a year from now, 5 years from now. What are the sort of stages you have to go through to make this real?
So our goal is to be on the market by 2025. The stages leading up to that are broken down at a high level. We have to finalize the design of the assay. We have to do a live patient trial for FDA approval. And then we have to go on and get our FDA approval to be even allowed to be on the market. There's so much more that happens alongside of that. We have to think of reimbursement and how insurance companies, are going to pay for this. We have to think about how we're going to drive adoption. So convincing doctors to actually use this on their patients. But from a technical point of view, we have a really early prototype at this point that we've tested out and works really well in a lab setting. And now we have to develop it to a scalable assay, so something that's actually going to be able to run on many labs across the world, get the clinical trial successful results, have the FDA approval, and then we can come to market.
So there you are, a woman sitting on top of this very scientific company. And I happen to know, because I do my homework, that your degree is in like economics. And
Yes.
how did that happen? How did you get here? I mean, if you're like, I came out of the lab and I'm the, toughest PhD in town, but how did you wind up doing this and getting to this place?
That is a great question. When I studied economics prior to, I would say, switching majors at Boston University, I was a biochem major and surely was going to become a doctor. That was my career trajectory. I come from a family of doctors, and so little did I know that really was not what I wanted to do, but I knew that I was passionate about the sciences. I grew up in the Middle East, and for the most part, career paths are very cookie cutter. You're a lawyer, you're an engineer, you're a doctor, and there isn't much in between, or there wasn't much in between, I should say. A lot has changed in the last 12 years. And when I got to college, I think I realized that there was so much more to having a career in the sciences than being a doctor or being in the lab. And it was my first job my internship when I was in my senior year, where I interned at a mid-stage diagnostics company also focused on women's health. And I really got a taste to understand what are all the other elements that go into patient care and developing innovation and bringing products to market. Yes, there are the people that invent them, there are the scientists, there are the people that study it, but there's the whole group of individuals that actually really work hard to ensure that these end up in the hands of doctors or in the hands of patients that actually have, and that this medical innovation has an impact. And so I ended up starting a career in women's health without even realizing that I would stay here my entire career, thankfully now. And then from there, it was kind of opportunity after opportunity at various different career steps, all in the healthcare industry. I worked in sales, I worked in business development, I spent a couple of years with QIAGEN working on a Bill and Melinda Gates project on expanding testing for HPV in East Africa. Phenomenal experience working with NGOs and really driving adoption for women to get tested in some of the most remote areas. I worked at a startup after that took a diagnostic from early development, licensed it out all the way to FDA approval and exit. And by the time I'd gone through all those experiences, I said, okay, well, I really want to stay in the space. I want to continue innovating. I want to find a problem and a solution that needs a connector. And so that's kind of where I came in, and I went on the hunt with my co-founders, and we looked at IP and we looked at patents. I think the statistic is 90% of medical innovation stays in academia. That's huge.
Right.
And so there are fantastic people developing who don't necessarily have the desire to commercialize. And then there's people like me that know and love to commercialize, but could not develop anything, even if I tried.
Right.
And so, that connection of how do we get that innovation into the industry, into the hands of the people that need it the most, is kind of where I come in.
So I'm curious, you were looking at all these things, and there's this professor at McGill with this capability. What was it about him or it that caused you to stop and go, wait a minute, this is the one? Or did you know it was the one, like love at first sight, or only after a whole lot of work back home did you realize it?
It was a mix. So we had been looking at a lot of things, endometriosis, just a variety of things. When we came across this, somebody had introduced us and said, I saw this professor at a conference and I think he's onto something. So we were connected with him, and he shared with us his data in the most transparent and honest way from a pure academic, this is interesting perspective.
Mm-hmm.
And we saw that data and said, if this data is true, and this was such a small sample set, if this data is true, there's something really interesting here, but why has nobody done this before? And this is the question that all of our investors have asked. Why has nobody done this before? And so from the initial look, we were very intrigued. And then we dove a lot deeper into it. We really started to understand, what were these targets? How had he run these experiments? How much could we rely on this data? Really started to understand more of the market. What were the challenges with ovarian cancer? And the more we looked into it and the more we worked with him, we realized that it was a really good fit for our expertise and our passion as well. Professor Zoghbi is really talented, but also a phenomenal collaborator. He is very transparent. He's excited about this project, and he works very closely with us, which really facilitates the relationship. We had been investigating a few other things where the relationship wasn't necessarily a match, but the technology was great, or vice versa. The relationship was great, but the technology was not necessarily that enticing. And here we found ourselves in a position where we had a phenomenal relationship with the scientist, and the technology was really astounding.
Amazing. So you mentioned when we were talking earlier that one of the key steps is clinical trial. And I was curious about that because in this case, if you get, your sort of standard 300 people and you run them through a trial, you need to find folks that have cancer.
Yeah.
And so, you could just have the wrong 300 folks. Good news, you're all healthy.
Yeah.
So do you actually have to run this somehow blind against samples from folks have cancer? Or does the population that goes through your clinical trial have to be enormous? Or what do you do to make sure that you can say, well, we found all the cancer in this group of women, which means you got to have women that actually have cancer?
Yep. So I would say it's a mix of that, actually. What we do with what's known as the inclusion criteria, so who do we select, who do we allow to participate in the trial, are only going to be women with signs and symptoms of ovarian cancer. So it's not sort of your broad everyday woman population, because you're
Right.
absolutely right. A trial that way would require 200,000 women to get to the prevalence for ovarian cancer. So we are starting with the population of all those women that have signs and symptoms of ovarian cancer. And from there, with our statistical analysis now, we've concluded that we need to run a trial of about 600 women. And from 600 women, we should pick up the number of cancer cases that are equivalent to the prevalence in the United States right now. So that means how often you actually see ovarian cancer among that group.
So what next? So the next year for you, market-wise, and the next year for you, investor-wise, if we come back and do this again same time next year, what do you think you'll be talking about? Where do you think the company will be at?
I am aiming this time next year, we'll have what's known as our assay feasibility and prototype complete. So taking this invention from the academic lab and having converted it into a scalable prototype now at this point. And we're actually planning to have our first I would say milestone and next step with the FDA as well. So the FDA has this classification called breakthrough designation, which
Right.
is for certain devices, diagnostics, or therapeutics where there is a huge need, it's a life-threatening disease, or there's a huge need and there's nothing on the market for it today. And so we're going to be applying for that by the end of this year. So by this time next year, we should know from the FDA what our classification would be when it comes to breakthrough. And then that will allow us to really start planning for the next step. So we have our prototype assay, we have our clearance with, or our understanding, I should say, with the FDA to move then into designing the clinical trial and moving on from there.
Got it. Sounds exciting.
Now that I'm on the record for it, you can ask me about it in a year.
We'll be back. We'll play this like one of those old Johnny Carson clips. Remember that? Remember that?
Remember that?
And see the joys of live television, as it were. Well, we'll come back next year and see where it stands. I mean, from our point of view, investing in you was easy. We look for big impact that's here, novel approach. This has never been done before. Real deep grounding in science as opposed to just kind of fly by night, and a great founder and team, and it was all here. So we're very excited about this. We can't wait to see what happens next, and we hope that the potential of this new approach is there. The whole notion of moving more and more toward prevention, early detection, away from crisis intervention and all those costs. I mean, apart from everything else, the cost of catching this early is a trifle compared to the cost in every respect of catching it late. Even if you happen to survive it, your life is wrecked and your job is wrecked and the costs are astronomical. And this is an entirely different, potentially an entirely different experience for the human being involved. And that's what we hope can happen.
That's exactly right. We're super excited to have all the Joyance team, supporting us and on this journey with us.
Same here. Well, we'll come back next year now and we'll have to see how it goes. Great to talk to you. Thanks for coming on.
Thank you very much for having me.